Hello, I'm Rozena Shirvani, and this is my colleague Asma Syed, and we are presenting the characterization of anti-cancer properties of the fungal metabolite Ophiobolin A. So Ophiobolin A, or OPH-A, is a secondary metabolite and a phytotoxin produced by the pathogenic fungi Coccobolus heterostrophus. Numerous studies have shown that the anti-proliferative effects of Ophiobolin A against a variety of cells, including bacteria and various cancers, including melanoma, glioma, and leukemia. Recent studies have shown that OPH-A induces peroptosis-like cell death in glioblastoma multiforme cells via vacuolization of the cytoplasm and enlargement of the mitochondria and endoplasmic reticulum. Notably, unlike apoptosis, peroptosis cell death lacks DNA fragmentation and activation of caspases, creating a possible mechanism for targeted treatment of malignant brain tissue of GBM patients who have GBM cells that are highly resistant to the pro-apoptotic treatments. So this is our inspiration for investigating more into this novel treatment for possible anti-cancer of GBM, which is highly resistant to current treatments and very fatal in current populations. So this study aimed to further characterize the effects of this promising anti-cancer agent on the survivability and cell morphology in glioblastoma cell lines U118 and U87, breast cancer with cell lines MCF and T47D, and neuroblastoma cells SH, SY, 5Y, and rat phemochromocytoma cells PC12. The lines of SH, SY, 5Y, and PC12 are serving as two popular neuron models to test the drug toxicity and variability that would be found in normal brain tissue neurons. And figure 1 is the chemical structure of OPH-A that we are investigating.
So I just wanted to talk a little bit about the reason why we wanted to investigate OPH-A. In previous literature, as you can see in this image that I'm pointing to with my mouse, there are hypothesized mechanisms for which this drug that we're investigating induces parapoptosis in glioblastoma cells as compared to apoptosis pathways. So our drug that we're investigating actually blocks this channel right here as hypothesized by previous literature, and that induces a downstream cascade, which causes an increase in intracellular calcium in cancer cells, mitochondria, and the ER, which we hypothesize leads to cellular swelling and vacuolization. So this is the proposed mechanism that may be the cause of the drug's potential anti-proliferative properties.
So for our methods, we use the cell lines MCF, which is the breast cancer, PC12, which is the pheochromocytoma serving as the neuron model, and U87, which is the glioblastoma cells. The other cell lines we plan to use were T47D and U118, and the cell lines during this experiment were non-viable and not published in this current data, and they're still being investigated at this time. So of those cell lines, we had a treatment group which received 1 micromolar of OPH-A, and we compared that with the control group that received the same volume of DMSO. So we observed with computer-assisted microscopy at 1, 3, and 6 hours post-treatment of OPH-A, and we compared the cell morphology, and then we used a cell counter to measure the cell survivability and cell death percentage. And we also compared this with serial imaging to analyze our results.
All right, so now I just want to talk a little bit about our results. So as you can see in our table A, B, and C, here are some images that we were able to produce from our research. So figure A shows MCF cells, which is our breast cancer cell line. PC12 is pheochromocytoma cells, which serve as our neuron model, as Rozena mentioned previously. And U87 is our glioblastoma cell line. So what we found in our research was that after the application of our OPH-A in our MCF cell line, let me zoom in so you all can see a little bit better. In this circle, we actually see an increased amount of vacuolization in our breast cancer cell lines. So if you can see here, there's actually an increased amount of basically white specks, which show signs of cell death just after 3 hours of application of a 1 micromolar amount of our drug. And also this vacuolization increases after 6 hours post-treatment as compared to our control, which is using just DMSO. And then for our PC12 cell line, which is again serving as our neuron model, as you can see in our control, we have healthy looking cells throughout 1, 3, and 6 hours. But in our 1 micromolar of the drug application, you actually see some more detachment of cells, so which means more floating cells. So our neurons here, for example, look healthy, but after 6 hours of application, there's actually more detachment, which is seen as circularization of the neurons as compared to like elongated neurons, which are healthy. So importantly, this does not necessarily mean that the cells are dying. It just means that they're, rather than being attached to the surface of the plate that they're being grown on, they're actually being like they're floating in the medium. And then for our U87 cell line, as you can see in our arrow, after 6 hours of drug application, we actually saw some elongation of our neurons, like both the body and also the projections, as compared to 6 hours of our controlled just DMSO. The DMSO still shows healthy glioblastoma cells, but the drug application shows some morphological changes that is seen in a time-dependent manner. So those are just our results from our images. Sorry, from our, just from imaging. And then here on the right, we continue into our results section. We also did a cell survivability analysis, and then we also followed that with analysis for significance using a 2-sample equal variance t-test. So in figure A, you can see that there was a statistically significant difference between our DMSO control and our OPHA-A application in our MCF cell line, which is our breast cancer cell line, showing some promising results for our drug. And in our PC12 line, which was our pheochromocytoma cells that again served as our neuron model, there was not a statistically significant difference in the drug group versus our control group, which is great because that potentially indicates that there is not a lot of toxicity associated with our drugs, meaning that there may be strong indication that this drug could be a safe drug potentially in animal models in the future. And then in our U87 group, when we were looking at cell survival rates, there was not a statistically significant difference currently in our research, but in previous literature, there has been significantly different, like there was a significant reduction in survivability in glioblastoma cells. All right.
So in conclusion, our study currently highlights the importance of understanding the mechanisms underlying the OPHA-induced cell death as a potential candidate for cancer therapy in cancers highly resistant to current treatment options. In previous studies, OPHA was found to induce cell paroptosis and decrease cell numbers in glioblastoma cell lines. And in this study, the MCF cell lines, which is breast cancer, shows statistically significant reduction in cell survivability post-OPHA treatment, and this change was not observed in the PC12 cells. So for our further direction, the researchers are currently planning to continue studying the anti-proliferative effects of OPHA in other cancer cell lines, including breast cancer. Additionally, the effect of OPHA in healthy cells will be investigated.
And that is all for our presentation. Thank you all for joining us, and that's all.